Hello, in this video we will understand about the biosynthesis of dopamine. The dopamine or dopamine. Basically, it is the neuromodulatory molecule as a neurotransmitter released from the nerve ending, the nervous system, especially in the brain. Organic chemical of catecholamine and phenylalanine, which that is the major composition from that. Dopamine constitutes about 80% of the catecholamine content in the brain. So the a rich amount of dopamine are present in the brain and also periphery, but just remember that in the brain. Let's begin to understand the dopamine metabolism. Itself is strongly linked to oxidative stress as degradation and generates reactive oxygen species which that damage the tissue also, and dopamine oxidation can lead to endogenous neurotoxin, whereas some dopamine derivatives show antioxidant effect even. So let's begin to understand of the interneuron, which that is involving the production of dopamine with the response of reward as the response of the pleasure and events, a lot of things which that is related to the happiness and release from the basal ganglia, which that is also involved for the decrease of the dopamine will lead to Parkinson disease. This is the nerves, which that is neuron contains soma, axon and terminal synaptic and the Schwann cell nucleus and dendrites. Let's begin to understand the dopamine biosynthesis, which is our target to understand in this video. So the L-levophenylalanine, which that will be convert into the L-tyrosine in the presence of phenylalanine hydroxylase enzyme to addition of the hydroxyl group. After this is the dopa decarboxylase enzyme will lead to decarboxylation to produce the tyramine. But anyhow, let's understand further process. The tyrosine or tyrosine hydroxylase enzyme TH will lead to convert this L-tyrosine into the L-dihydroxyphenylalanine, L-dopa. L-dopa is important is the drug treatment can be introduced for the Parkinson disease to cure. The dopamine will convert due to the dopa decarboxylase enzyme to produce the dopamine. Inside is the naturally the biosynthesis of the dopamine by this and tyramine also in the presence of CYP2D6 gene. While this MAO enzyme will lead to 3,4-dihydroxyphenyl acetaldehyde is the POPAL, which that produce this is the an ALDH enzyme used to produce the POPAC, the acetic acid, basically the dihydroxyphenyl acetic acid. While dopamine also convert into the 3-methoxytyramine, tyramine, which that convert into the presence of the MOA enzyme will lead to 3-methoxy-4-hydroxyacetaldehyde formation, while the COM T is the catechol O hydroxymethyl transferase enzyme used to produce the homovalenic acid. This homovalenic acid is the degradation of the dopamine by this process, and this dopamine degrade and decrease, and 3-methoxy-4-hydroxyacetaldehyde formation. And so we have discussed about the dopamine biosynthesis and degradation. So let's begin to understand the major mechanism. This is a synaptic nerve and terminal and dopaminergic neuron are present which that target to the different tissue, different cell with the different receptor like D2 and D1 receptor. And when target will lead to response in brain or periphery, remember that things. So this is the biochemistry of the dopamine, the biosynthesis of the dopamine. I hope you make sense about that. Let's begin to understand the family and receptor, the gene and types as well as mechanism. This is a short video for understanding the biosynthesis of dopamine, but additionally I am mentioning this graph. D1 like is the D1 and D2 receptor. Family is the D1 like dopamine. So that gene is the DRD1, DRD5, and the G couple protein, GS couple protein receptor use increase the intracellular level of cyclic AMP by activating the adenylate cyclase. While the D2 like is the D2, D3 and D4 is a receptor, while the gene is DRD2, D3 and 4. And the GI coupled protein will lead to decrease the intracellular level of cyclic AMP by inhibit the adenylate cyclase. In this way, the preparation and the synthesis will occur of the neuron. And the tar is the tar1 and the tar1 trace amine associated receptor 1, which that involve the GQ coupled and S coupled protein receptor will lead to increase the intracellular level of cyclic AMP and intracellular calcium concentration. It means it will be progress and will lead to get the response, and that is why both function. The synthesis of the basically is the neuroplasticity and the synaptogenesis is occur due to the dopamine. Remember that things. So that is why the dopamine will lead to development of the brain as well as the memory, memorization in our brain long term. The long term potentiation will occur. By this several function we will discuss in the next video, because this is the series of the nervous system we are understanding. So next video we will learn about how dopamine will work with a different receptor. So I hope you make sense about this biosynthesis of dopamine. Next video we will understand about the mood of action. Thanks for watching. Bye.